Good evening, everybody, and thanks for the introduction, and thanks to all of you for coming. So I'm a researcher at Memorial Sloan Kettering Cancer Center, and I'm going to talk to you about a topic that will be very dear to everybody, to you, which is food and nutrition. But I want to explain a bit how not we as human beings are eating, but how the cells in our body are acquiring nutrients and what they're actually doing with it. So in principle, you can think about a cell like something like an engine, for instance, like a car. And so in order to build a car, you need building blocks to construct the car. But then once you have the car, you need a lot of energy to keep it running. And that's very similar actually to a cell, that you need building blocks to construct a cell. But then the cell contains a lot of machines and pumps and different components that require energy. For instance, if our muscles are working, they require a lot of energy to produce force. Of our neurons and brain cells, they require a lot of energy to transmit electrical signals. It's one thing that we are very interested in is how cells can use their nutrients for these different processes. For instance, how to decide that a cell makes more cells, or it uses the nutrients rather to produce energy and then to do work. And that's also, again, similar to cars, right? Like if you have a car manufacturer who wants to build cars, then what's really important are building blocks and not so much energy. I mean, you may need some energy to construct the car, but it's really more material and not so much fuel. So Cozy Science asked me to not bring any visual support, which I find pretty challenging. And fortunately, almost everybody of you has some visual support here, which is beer, which is a very good example to explain some basic principles of how cellular metabolism works. So we know a lot of the biology of beer brewing from the French microbiologist Louis Pasteur, who worked on this in the 19th century. And so one reason actually why Pasteur wanted to work on this, and he mentioned that in a letter to a friend, is that at that time the relationships between France and Germany, where I'm from, were not particularly good. And so he felt that if he were to work on beer brewing, and if France then would overtake Germany in terms of beer brewing technology, that would be a very major blow to the German people and psyche. And so that was one of his motivations to explore this. And so now what happens in beers or in fermentation in general is that you have a liquid that is very rich in sugars, and then you put yeast in, and yeast turns sugar into alcohol and excretes it. And that's what makes our beverages alcoholic. But the other thing that happens is that the yeast proliferates, and actually the main thing is it doesn't want to make alcohol, it wants to proliferate. And if nutrients are very abundant, and in particular if the cell has a lot of sugars, it's a very effective way to produce mass by turning it into alcohol, and at the same time to produce biomass. And so the main task that Pasteur set himself, he actually failed in, because he wanted to find a way to make yeast produce alcohol and do fermentation without growing. Because if yeast grows, it consumes parts of the beer ingredients, and the process gets more ineffective. And so if you had a yeast that did not grow but just ferment, you would be much more effective in the materials that you have to use for brewing. And he never managed to do this. So if yeast produce alcohol, it always also proliferates. And so the main reason why it produces alcohol is to actually produce energy for proliferation. There are also some fringe benefits for the alcohol production. For instance, it kills almost everything else in the environment of the yeast. And for instance, our cells would die if they were exposed to the amount of alcohol that a yeast cell can produce. But that's really more a side product. And so the behavior of yeast changes a lot if one lets the culture grow for a longer time, because then nutrients become more depleted. And then the yeast actually takes up the alcohol again and oxidizes it and burns it to produce energy. So this is already a good example for how cells can use nutrients for very different means. Yeast can take sugar, ferment it, secrete as alcohol and produce a lot of biomass and make new copies of yeast. Or it can take up the alcohol and burn it to produce energy and just survive when nutrients become limited. And so yeast is a unicellular organism, and in principle almost all unicellular organisms on the planet do exactly the same thing. If a single cell organism has food, it makes copies of itself. And it does so until the food is gone, and then it, for instance, in the case of yeast, it may produce spores, or it may enter some kind of dormancy or so, and try to live until it gets a new food source, and then again it makes copies of itself as long as the food is there. And so now if we move to animals, which are very complex assemblies of lots of different cell types, like human beings, for instance, consist of maybe 100 trillion cells. there we run into a big problem because our blood and body fluids are very nutrient rich, and so our cells are present in a very high nutrient environment for all of their life. So we as human beings, of course, may be starved sometimes, or we may be hungry, but our body fluids are always nutrient rich, and our body does a lot to ensure that that's the case. Like, for instance, we start to mobilize glycogen in the liver, or then we may burn fat, and at some point we start to degrade muscle proteins, and all of that serves to keep fairly constant nutrient levels in our bloodstream. And that is important, for instance, for the brain to function, and if nutrient levels drop too much, then we get unconscious. So like I told you, like unicellular organisms, if they have nutrients, they proliferate. And now our cells have nutrients all the time, and so they must not behave as yeast does. And in general, it's a very central process in the evolution of complex animals that cells stop behaving like a unicellular organism. And so what happened in evolution is that our cells lost the ability to proliferate in an autonomous way. So a single cell in our body cannot decide by itself to grow. And that's very different from bacteria or yeast or other organisms. And so what instead happens is that our body produces signals that tell a cell proliferate. And that is important so that this happens in a controlled way. Just as a thought experiment to illustrate this, if every cell in our body were to just divide once at this moment, then we would turn into a monster. And it's of course important that proliferation can again be activated because, for instance, our skin reproduces at a low rate, and then if we get a sunburn, like the dead skin cells fall off our body, and then like skin stem cells have to proliferate at a higher rate to replace the damaged tissue. Or if you have an infection, then immune cells proliferate a lot to produce cells that fight the pathogens. And so the most important thing for cells to make copies of themselves and to proliferate is, of course, that they acquire nutrient. And so what people realized over the last maybe 10 or 20 years is that the signals that instruct our cells to proliferate directly tell our cells to take up nutrients. And so this is one way how our cells can be in an environment that always has a lot of food and a lot of nutrition and not eat it. And the body has to tell it to eat it. So for instance, if you were to again take a yeast cell and put it into a dish that's in a very nutrient-rich like medium, it would proliferate until the medium is gone. If you take a cell from our body and put it into the same medium, it would not do anything. It would not take up any food, and it would starve to death. And it would do so even though the nutrients would be present in the environment. If we now take a growth signal, what the cell would do right away is it would take up nutrients and would use them to produce biomass and start to make copies of itself. And so one of the signaling molecules that does this you will be very familiar with, and that is insulin. Like, for instance, if we take a rich meal, our pancreas senses, okay, we have a lot of nutrients, and it makes insulin. And insulin is one of these central molecules that tells cells take up nutrients and store them for a rainy day. And so there's some hierarchy in our body, like the liver gets the first part of taking up nutrients after we eat, and then our fat tissue takes up nutrients, and our muscle may store nutrients. And once all of these cell types have eaten, other cells take up what's left of the meal. And then our blood nutrient levels go down again, and insulin goes down, and then our cells stop eating and just survive with the nutrients that they have just taken up. And so this is a central mechanism that allows that our cells behave socially and that they don't, in an uncontrolled way, just burn all the energy that is present in our body. And so now the question is, of course, what happens if this goes wrong? And one case where this can go wrong is, of course, if we have an infection of a foreign organism, like if we are infected, like if a pathogen such as a bacterium or yeast comes into our body, it finds itself in an incredibly nutrient-rich environment. And normally we have an immune system to fight this, right? So normally infections stay very controlled and very localized, and our body manages to fend off this invader. But then if you look at immunocompromised patients, such as patients who have undergone chemotherapy or have done organ transplantation and thus take immunosuppressive drugs, if these people get a yeast infection, the yeast proliferates very rapidly and takes up all the nutrients that it finds in our blood, and the patient dies very quickly because of energy depletion. So this is now, of course, an example where we have like an external cell that exploits the high nutrient levels that are present in our body, but a context where this happens to our cells is the development of cancer. because cancer is a disease in which cells start to behave very selfish again and behave similar to single-cell organisms, and they regain autonomy about proliferation, and they also learn again how they can eat themselves without our body having them to tell how to eat. So one cancer experiment, or like one experiment that illustrates certain aspects of cancer, something that many of you will have done accidentally, and that is if you just forget like a slice of toast somewhere, or just have bread that gets old, and so at some point, just stochastically, individual spores of yeast or mold will land on this bread, and so these spores now find themselves in an incredibly nutrient-rich environment and start again to make copies of themselves and proliferate. And I'm sure you have noticed that these mold colonies on bread are circular, and the reason for this is that it's one single cell that then grows in a radially symmetric way to the outside. And but so at some point the nutrients are gone, right? And for instance, if the cell lands more to the edge of the toast, then the nutrients will run out quicker because it just hits the edge. And so what it then does, it starts to invade the surrounding bread, or it produces spores that then can be transferred to other parts of the bread. And so what you see is that you often have one initial colony that grows, but at some point you see colonies popping up all over the toast. And that is very similar to what happens in metastasis, so that you have a cancer that grows slowly in a restricted way in an organ, but at some point the cancer cells learn to leave the environment. And there's actually a lot of evidence that starvation is contributing to this, and that the cancer cells try to go to an environment which is rich in food, and then they start to colonize other organs and grow in this environment. So that the insight that cancer is a metabolic disease is actually fairly old, and so the first person who put forth this concept was the German biochemist Otto Warburg about 100 years ago. And what he saw is that cancer cells take up huge amounts of glucose and secrete a lot of lactic acid, and they do so at a much higher rate than surrounding tissues. And so this is something that you also may have heard of, because the way that many cancers are being diagnosed by now is by PET imaging, where one takes a molecule of glucose that's labeled in a certain way so that it can be detected by PET imaging. And then if one injects this into a patient, then there are a couple of organs that light up, for instance, the liver and brain, which are organs that consume a lot of glucose. But otherwise, almost all cancer types very quickly light up with glucose. And so in contrast to the organs in which they arise, they start to just eat huge amounts of sugar in a very uncontrolled way. So the idea that cancer is a metabolic disease is fairly old, but then people discovered DNA and people started to discover cancer mutations, and so cancer biologists forgot a bit the older findings that it's actually a metabolic disease, and worked a lot on proliferation per se, and how far mutations that arise during cancer progression regulate cell proliferation. And so one of the really big insights in the last 10 years or so, and that's something that we actively are studying right now, is that all the oncogenes that arise early in tumors and are the first genetic lesions that we can detect in cancer, all of them instruct cells to take up glucose. And so this already tells you that one of the reasons why these are probably selected for in cancer development is because they start to allow cells to eat in an autonomous way and not have to wait for the body to tell it to do so. And so now these cells start to take up a lot of glucose, they start to eat. These mutations also start to reprogram the metabolism in a way that the metabolic rate increases. And what happens then is that cells are very resistant to death. I mean, like I told you, like if you were to take a cell out of our body and put it into a dish, it would die if no signal told it to eat. And we think that that also can happen in our body, that if cells are neglected and they don't get a signal, take up nutrients, they die. And this could actually be a mechanism against, like, to protect our body against cancer. But so now that the cells have learned how to eat in an autonomous way, they're pretty protected from death because they know how to get the energy by themselves. And the faster metabolic rate actually increases reactive oxygen species, which can damage DNA and increase mutagenesis in the cancer. And so what happens next is that the cancer that is overeating the cancer cell and has a high metabolism starts to mutagenize itself, and that allows it to acquire further mutations that drive cancer progression. And so when people realize this, there's a lot of interest right now to develop drugs that really target specific mutations in cancer. And the benefit there is, of course, that this allows to target the cancer cell itself and not other cells in the patient. And so one of the things that people are trying to do right now is that they don't block proliferation per se, but they block nutrient uptake in the cancer cell. And this would not hurt so much the surrounding tissues, which need much less nutrients because they don't build much biomass and they don't proliferate much. But it could acutely hurt the cancer cell, which has a high metabolic rate and is actually addicted to this nutrient supply. So the other side of this coin is, of course, the impact of nutrition itself, right? There's a very clear correlation between obesity and cancer development. And it used to be that tobacco is the #1 preventable cause of cancer, but now most countries, like smoking, has gone down a lot, and so the cancer, like lung cancer and other cancers that are produced by smoking, have decreased very substantially. Another preventable cancer is skin cancer, and now that we use much more sunscreen and try to not be in the sun in an unprotected way all the time anymore, skin cancer is also going down a lot. And so I think like maybe 5 years ago, obesity-related cancers overtook these other causes in the #1 cause of preventable cancer. And so the mechanisms are still not entirely clear, but one reason why we think that obesity can actually increase cancer is because now you have a very nutrient-rich environment again in the body. And so if we eat more food, the first thing that we do is we get larger. Like human beings used to be smaller 100 years ago, and they used to be even smaller 200 years ago. And that's very clearly related to diet. And so now we eat more and we have a healthier diet. And so we just grow more and build more and stronger healthy tissue. What happens then if we eat even more, which started to happen maybe 50 years ago, is that we start to store excess nutrients in the liver and in adipose tissue. And there they are tucked away from other cells and stored in a fairly safe way. But if the storage capacity of these cells is exceeded, what happens then is that a lot of other cell types in our body get much more nutrients than they ought to. And so this then again allows cells that acquire mutations to survive and to mutagenize themselves and to turn into cancer cells. So we think one reason why cancer is related to overeating and obesity is really that cells have more nutrients than they should have, and the protective mechanisms that regulate nutrient uptake are out of place. So the last thing that I want to briefly touch on is that like initially there was a lot of interest in how cancer cells use sugars, and then people also started to investigate how cancer cells use amino acids. And by now there's a lot of interest how our cell types use a lot of different other nutrients. And that is, again, best illustrated if we go to unicellular organisms. Like if you, for instance, have a yeast cell that sits on a rotting fruit, it has a huge amount of sugar that it can use to build biomass. But if you're an amoeba or some other protist that lives in a pond or in a lake, there are really no free sugars or amino acids there. And so these cell types have very different ways to acquire food. And a lot of these cells are predators. So there's some pretty amazing videos that you could look up on YouTube where you have amoeba that chase bacteria, and it's really one single cell that chases another cell, and the bacteria try to escape and they move around, and the amoeba follow the direction, and at some point if they catch up they swallow the bacteria and eat them. And a lot of other cells are scavengers and take up not whole cells but maybe like cellular debris or rotting material or other things, and live on these kinds of foods. And cells do so by taking up these macromolecules and cells and then trafficking them to intracellular organelles that basically function as cellular stomachs. And these cellular stomachs really function in a very similar way as our stomach. They acidify, they secrete enzymes that are very active at this acidic pH, very similar enzymes to those that our stomach and intestine produce, and there the nutrients and cells are degraded, and this creates an intracellular source for amino acids and sugars that the cells can utilize. And we call these organelles lysosomes because they lyse the material that they take up. And so there are some professional cells in our body that do the same, and these are mostly immune cells, because one way to get rid of pathogens that invade our body is that one just eats them. And so we have various immune cells that just go around in our body, very similar to amoeba in a pond, and they just try to find bacteria, and once they find them, they eat them. But these pathways are also present basically in every cell type. And so something that we've been starting to appreciate over the last couple of years is that cancer is much smarter than we would have anticipated, and it does not only rely on amino acids or glucose or so, but it can also reactivate these eating mechanisms by which it swallows surrounding materials or proteins that are present in our body fluids. And you can actually see in tumors that sometimes cells completely eat other cells, and so you have something like a survival of the fittest, by which certain cells eat their surrounding cells and can proliferate and use these nutrients to survive. And one reason why these eating strategies are important in cancer is because cancer often grows faster than the vasculature, and so cancers often start to develop a very poor blood supply, and then they start to explore these different ways of acquiring nutrients. And these are particularly aggressive cancers because they now have a switchblade of a lot of different ways to take up food. They can take up glucose and different amino acids, so they don't forget to take up these low molecular weight nutrients, but now they start to be able to also take up cellular debris or proteins or other cells or many other things in their environment. And so when we now try to inhibit nutrient uptake, we realize that in some cancers it's not as simple as blocking one specific way, but one would have to block a variety of different ways of nutrient uptake. And so something that we're actually very excited about right now is that we try to not prevent cancer cells to overeat, but that we actually use their overeating to poison them. I mean, like I said, we already use the overeating to detect them. I mean, we take these glucose PET molecules and can very well detect cancer because it's overeating glucose, and we can see it in a PET scan. And similarly, we think that we can take cancers that take up a lot of macromolecules and cellular debris and use that as a way to deliver poisons that normal cells would not take up, but cancers now do. And so this would be a way to selectively kill cancer cells. And that is something that we are exploring right now in the lab. So yes, so with that I would like to conclude. So like I said, one of the fascinating things when we study our bodies is that our cells really have to be retrained in a lot of ways from our unicellular ancestors because they cannot just proliferate in an uncontrolled way. And the major difference that we actually see in the behavior of our cells, in the behavior of yeast cells and bacteria, is that our cells have forgotten how to eat, and they're instructed in a very regulated way how to take up nutrients. And these are important control mechanisms that allow our bodies to function. And this is actually something that we can actually probably learn from our cells, that we should not only eat if food is available, but we should maybe in a more controlled way to learn how and when to indulge. Thank you.